And first of all, thank you again for joining today. I will briefly introduce the topic of this seminar and the group of people that will provide the talks today. And then I will leave the stage to the first speakers, and the seminar will be organized with an initial part of presentations, and then we will leave some room at the end for questions and potential discussions that may arise. Okay, let me share my screen. Okay, you should be able to see my screen, and okay, you should be able to see the first slide of the presentation now. Okay. Yes. Okay, as you know, today we will discuss experimental and computational methodologies centered around the application of bone regeneration. And in particular, we will focus on in vitro models and the experimental and computational methodologies to improve and use in vitro models for basic and applied research. People that will participate to this seminar today will be Professor Luca Munaron and Dr. Giorgia Chinigò from the University of Turin, Professor Gionata Fragomeni from the University of Catanzaro, Engineer Riccardo Smeriglio, Dr. Roberta Bardini, which would be me, and Professor Stefano Di Carlo from Politecnico di Torino, and Professor Department of Control and Computer Engineering, and Professor Tommaso Lorenzi and Dr. Nadia Loi from the Department of Mathematical Engineering of Politecnico di Torino. So the seminar will have this outline. We will start introducing the biological complexity of bone regeneration. And this part will be provided by Dr. Giorgia Chinigò, who will also present the second part about experimental methods to study cellular crosstalk in bone regeneration. So in the initial part, you will be presented with the challenges that biological complexity poses in this use case, and the experimental methods from the biological laboratory that are in place, are the frontiers method to tackle this complex problem. Then we will move to the analysis of models based on bioreactor designs. In fact, Giorgia will talk about more static in vitro models. Professor Fragomeni otherwise will talk about dynamical models, so bioreactor and dynamical culture systems. People from Politecnico di Torino, so Professor Lorenzi and Dr. Loi, and Engineer Riccardo Smeriglio will talk more about mathematical and computational approaches to study in vitro model simulation, design, and optimization. And then we will end up with a focus, which I will provide, about the importance of interdisciplinary collaboration in tackling this kind of complex problems, taking to you an example, which is the Saisei project, a collaborative and interdisciplinary project some of us are collaborating to develop, and which, in our opinion, is a good use case of the synergistic merge of these different approaches to in vitro systems. Let me also thank Professor Catapano, who was the one inviting us to give this seminar for the Osteonet Consortium, and who will join probably later during this seminar. So I will leave the stage to Giorgia, to Dr. Chinigò, who will introduce you to the biological challenge. And I take the chance to also introduce the second talk that she will give in continuity with the first, experimental methods to study cellular crosstalk in bone regeneration. So, Giorgia, you should be able to share your screen.
Okay, thank you, Roberta. I'm trying
to you. And I take the chance to invite all of you to ask questions at the end of the specific parts of this seminar, when the speaker finished. In those in between times, you are free to ask questions and to spark discussions if you deem it relevant. Otherwise, there will be time at the end of the whole seminar. Thank you.
Thank you, Roberta, for your kind presentation. Can you see my screen, my first slide?
Yes.
No. Ah, yes, okay. Okay, so first of all, good afternoon to everyone. My name is Giorgia Chinigò, and I work as a postdoc in the Turin Cell Physiology Lab under the supervision of Professor Munaron, so at the University of Turin. As Roberta mentioned before, my presentation can be divided into two. In the first part, I will give you a general introduction on the topic of this seminar. So I will speak about the challenge of complex intercellular interactions in bone regeneration. In the second part, I will more specifically focus on a challenge that is of interest in our lab, that is about bone vascularization. And so I will tell you about cellular model and cellular assay that can be used, that we use in our lab in order to study the crosstalk between endothelial cells and osteodifferentiating stem cells.
Giorgia, sorry to interrupt. We see your screen, but we don't see the full screen presentation. Only the anteprima.
Why? Let me check. I try to share.
Thank you.
So you see the screen of PowerPoint, not
yes.
the full. Interesting. Why? If I do like this. Now, again, it's the same.
Try to share again.
No screen at all.
I'm sharing.
We don't see the screen.
Okay. I try to connect to a second monitor, but probably I will be not able to hear you. But maybe we solve the problem.
No worries.
Now, does it work now?
I don't see your screen.
No shared screen here.
Neither.
Maybe try to stop the sharing and restart it.
Yeah, I try. I completely ask, okay, and retry from this. Sorry, schermo intero. I didn't select, okay, just select PowerPoint, not the entire screen. So maybe now it should work.
Perfect.
Now
we see the modalità presentazione now, but if you swap the screen,
Presenter mode. Yeah.
Yes.
Yes. Presenter mode. Okay.
Presenter mode. Okay.
Yeah.
And so I ask from this.
In the top bar, just in the middle part, middle impostazione.
Yes.
And there is one to invert it. No, the button to the right.
Postazione di visualizzazione. Postazione di visualizzazione in alto.
Yeah, that's
okay,
fine.
Perfect.
Okay.
Thank you.
Sorry.
No worries.
Okay.
Good to go.
Okay. So we all know, no, sorry, but there is something that is not working on my screen because the mouse is not working. My God. Okay. Okay. So we all know, can you hear me? Yes?
Yes.
Okay. We all know the complexity of bone tissue because bone is highly dynamic tissue that continuously undergoes constant changes. Its functionality, sorry, okay, mainly relies on the activity of three different cell types that are, on one hand, osteoblasts, that are responsible for bone formation through the deposition of unmineralized extracellular matrix rich in collagen and proteoglycan, so collagen type 1, osteopontin, osteonectin, for instance, as well as enzymes that are associated with the mineralization of the matrix, like the alkaline phosphatase. On the other side, we have osteoclasts that are responsible for bone resorption, a process in which osteoclasts secrete enzymes that are able to digest the old bone matrix and aged cells. In the middle, we have osteocytes that are fully differentiated osteoblasts responsible for the mechanosensitivity and mechanotransduction. So basically, osteocytes are able to detect mechanical stimuli and to transduce them into biochemical signals able to regulate the activity and the differentiation of both osteoblasts and osteoclasts. So bone health and homeostasis require a fine balance, let's say, between bone resorption and bone formation, the so-called bone remodeling, that play a central role in bone functionality, also because through bone remodeling, our body continuously restore old damaged bone for daily physical load and aging effects. And moreover, bone remodeling is fundamental for bone repair within 3 to 6 months more or less when a fracture, a healing occur. Indeed, one of the main features of bone is its great self-renewal ability. The bone healing and regeneration process is similar to the embryonic bone development process. It involves three main phases that you can see in this slide. In the first phase, inflammation, a blood clot is formed at the injured site, and monocytes, macrophages, and other phagocytic cells start to induce an inflammatory response. At the same time, mesenchymal stem cells, in order to balance the response, start to secrete some anti-inflammatory factor in order to avoid an excessive inflammatory responses. In the second phase, that is the repair phase, the so-called bone callus is formed. How? Because basically mesenchymal stem cells don't differentiate just into osteoblast, but also into chondroblast. And so the chondroblasts start to build an external structure at the injured site, that is bone callus, in which osteoblasts start to proliferate in order to close the clot. In the third phase, the remodeling phase, as you can see there, we have the osteodifferentiation of osteoblasts that start to proliferate and osteodifferentiate, as well as the activity of osteoclasts, so the establishment of the balance between bone resorption and bone deposition. Concomitantly, as you can see there, angiogenesis, that is the formation of new blood vessels, occur in order to well vascularize the new tissue. So bone has a great self-renewal potential, but however, when the fracture exceeds a site's boundary, for instance, after a severe trauma or congenital deficiency, osteoporosis, or other bone diseases, surgical intervention is required. Typically, the main procedure carried out in this case is based on the use of bone graft that can be directly derived from the patients. And in this case, we speak about autograft, or from compatible donors. In this case, we speak about allograft. But this procedure suffers of some drawbacks. Indeed, beyond to being expensive and of course invasive, because it is required to collect from the patient bone fragments, typically from his iliac crest, and beyond, in addition to the possible incompatibility when using allograft, one of the main concerns of this procedure is the potential for donor site morbidity. So the connected complications like including bleeding, neurological disorder, pain, the need to reoperate the patient, and so on.
Giorgia, sorry to interrupt again, but there is a banner in overlay. You should just click on NASCON. Okay, so the slide is fully visible. Thank you.
Sorry. And moreover, this procedure can solve just smaller site defects. Why? First of all, for the low amount of bone graft, and secondly, because its efficiency can be strongly compromised by the short supply from the surrounding tissue. That's why, in order to overcome these limitations, these drawbacks, in recent years, bone tissue engineering has emerged as an ever-increasing area of interest, also because bone is the second most transplanted tissue after blood. Which is the purpose of bone tissue engineering? Basically, to design and develop artificial bone graft, bone implants, to treat bone injury for the long term. The big challenge is to produce biomimetic scaffold that are able to reproduce as efficiently as possible the complex intercellular interaction that occur physiologically. So basically, as I said before, the inflammation process, the stem cells recruitment, and then osteogenic differentiation. Because the main goal, the main aim of bone tissue engineering is not only design some implants that allow good clinical outcomes, but above all, long-term efficiency for long-term repair. And this is a big challenge. In this slide, I summarize the main strategies exploited by bone tissue engineering that can be divided into two main categories. On one hand, we have the so-called endogenous strategies that plan to induce osteogenesis by exploiting the response of endogenous cells, basically by using scaffolds that could be made from a large variety of biomaterials, from inorganic biomaterial like metals and bioceramics to natural and synthetic polymers, as well as the combination of the two. The physical properties of the scaffold could be changed and further enhanced through the addition of surface modification, as well as the addition of soluble molecules like proteins or growth factor that play a central role in the osteogenic process. Another endogenous strategy is the so-called macrophages polarization, in which basically, very briefly, we use a scaffold in order to induce the polarization of macrophages 1 that are pro-inflammatory to the anti-inflammatory macrophages 2. The aim in this case is to improve as much as possible the osteointegration of the scaffold in vivo. The second category include strategies that plan to induce osteogenesis by the introduction of exogenous cells, basically mesenchymal stem cells, which I'll tell you about shortly. So in this case, it is easier, let's say, to ensure a long-term efficiency of the scaffold, because we are introducing into the patients some cells that physiologically play a central role in promoting osteogenesis by the secretion of cytokines and growth factor. For this reason, sometimes just the secretome of mesenchymal stem cells is introduced into the patients. Moreover, the overall efficiency could be further increased by introducing some genetic modification within mesenchymal stem cells before their expansion in vitro and then transplantation. And finally, the use of bioreactors could help in better control the physical environment in which the scaffold works, but our colleague from the University of Catanzaro will tell you more in detail about bioreactors in the next presentation. So it is clear that to obtain high performance biomaterial for bone tissue engineering, a multidisciplinary approach is fundamental, because several aspects should be taken into account. Not only the structural features of the scaffold, but also its physical characteristics, as well as mechanical properties, and finally, the biological features of the scaffold. For instance, an ideal scaffold should be, of course, biocompatible, but it should also be able to form a chemical bond to favor its osteointegration. It should have an interconnected porous structure in order to favor the homing of the cells, and also a surface that is suitable for cell attachment. So basically, to obtain the best results in terms of performance of the biomaterial, several expertise should be combined to each other. For instance, engineering, physics, and biology mainly. From the biological point of view, because I'm a physiology, these are the main aspects that should be taken into account when evaluating new biomaterial for regenerative medicine. First of all, the osteogenic differentiation of the cells. Secondly, the inflammatory response induced. Third, the so-called homing of stem cells, so their ability to be, the ability of the scaffold to provide a suitable surface, a suitable three-dimensional structure for homing the cells, the mineralization, and finally, the vascularization. In this regard, one of the main challenges in bone tissue engineering remains to obtain an adequately vascularized scaffold. The formation of an efficient vascular network within the scaffold is indeed necessary not only to support its proper osteointegration, but also its osteoinductive potential, and so to favor the osteogenic process. Bone is indeed a highly vascularized tissue. And in this case, endothelial cells, the vessels, are not only necessary to provide, oxygen and nutrients, but also to support, to sustain bone development and maintenance. Indeed, during bone development, as well as during bone regeneration after a fracture, angiogenesis, that is, as I said before, the formation of new blood vessels, precede osteogenesis, as you can see there, and determines its efficiency. In this regard, not surprisingly, it has been shown that genetic models with bone defects are also associated to defective vasculature, thus supporting the central role of bone vascularization in the context of bone regeneration. But how does vascularization impact on osteogenesis? As you can see from this slide, through the secretion of a large variety of soluble factors that are called angiocrine factors. As you can see there, indeed, the endothelial cells are able to secrete some factors that play a central role in all those processes I spoke about at the very beginning of this presentation to describe bone functionality. For instance, endothelial cells help in supporting the survival of osteoprogenitor cells and hematopoietic stem cells that are predetermined to differentiate into osteoblasts, but not only, because as you can see there, endothelial cells are also involved through the secretion of these angiocrine signals in the osteoblast-mediated osteogenesis and in the osteoclast-mediated bone resorption. At the same time, bone cells itself, through the secretion of the so-called angiogenic factors, stimulate the recruitment of new blood vessels, thus establishing a real coupling, let's say, between osteogenesis and angiogenesis. One of the main actors of this coupling is, of course, the well-known vascular endothelial growth factor, VEGF. As you can see from this cartoon, very briefly, osteoblasts secrete VEGF, thus stimulating the proliferation and migration of endothelial cells. Endothelial cells, in turn, in response to VEGF stimulation, secrete some factors like the bone morphogenetic protein 2 and 4 that play a central role in mediating the osteodifferentiation of osteoblasts. Moreover, bone osteoblasts and endothelial cells, in response to VEGF and in a dose-dependent manner, control the expression of this protein, that is semaphorin 3A, that play multiple roles. On one hand, promote osteodeposition by osteoblasts. On the other, inhibits osteoclast differentiation and so bone resorption, and moreover, favor the stabilization of the new vessels by favoring the recruitment of this specific class of monocytes that are neuropilin-1 positive. So it is clear that there is a strong coupling between the two processes, and that a better understanding of the molecular mechanism underlying this coupling could help in developing scaffold that are able to support a proper the formation of a proper vascular network, so improving, let's say, the outcome of bone tissue engineering. That's why in our lab, we decided to deepen the molecular physiology of this crosstalk between endothelial cells on one hand and osteodifferentiating stem cells in the other. To this purpose, we look at mesenchymal stem cells because, as you know, mesenchymal stem cells physiologically play a key role in bone regeneration. Indeed, when a fracture occurs, mesenchymal stem cells actively participate into the inflammatory phase, but also into the repair and remodeling phases we spoke about before. In the recent years, mesenchymal stem cells gain great interest in regenerative medicine because of their peculiar features. Mesenchymal stem cells indeed have a high proliferative capacity. They have also a self-renewal ability, and lastly, but not least, mesenchymal stem cells are multipotent stromal cells. It means that they could potentially differentiate into a wide range of different cell types, including the one we are interested in, so osteoblasts, osteocytes, chondrocytes, and so on. Mesenchymal stem cells can be isolated, as you can see there, from different tissue, the oral cavity, bone marrow, adipose tissue, umbilical cord, and fetal dermis. And the idea is basically to isolate these stem cells from the tissue and then expand them in vitro and then apply them in clinical trial, transplanting them into the patients. In our studies, we use two main types of mesenchymal stem cells. First of all, stem cells isolated from the adipose tissue, thanks to a collaboration with the Dental School of Torino. As you can see from this scheme, the adipose-derived stem cells can be induced to differentiate into osteoblasts by simply adding some specific factors to the cell culture media, such as including ascorbic acid, dexamethasone, and beta-glycerophosphate. Then the osteodifferentiation of ASC is driven by the upregulation of this gene, RUNX2, that for this reason can be used as a marker of their osteodifferentiation that ultimately results in the expression of some genes that are strictly associated with bone deposition, like the alkaline phosphate, as I mentioned before. But another abundant source of mesenchymal stem cells is the oral cavity. And in this case, thanks to their different anatomical origin, these stem cells could be more effective than the previous in treating, for instance, dental defects. That's why, again, in collaboration with the Dental School of Turin, we isolated stem cells from the dental pump. And we use this shed to induce their osteodifferentiation. Among the advantages offered by this kind of stem cells, there are their high expansion capability, their great differentiating potential, as you can see there, but also a strong immunomodulatory activity. And moreover, these cells are quite easily to obtain, to isolate, and then manipulate. So, as cellular models, we use these two kinds of mesenchymal stem cells, one from the adipose tissue and the other from the dental pump in the oral cavity. And what we did was to, on one hand, keep these stem cells in a basal growth medium that is able to maintain them in their staminal phenotype, and contemporarily putting these cells into an osteodifferentiating medium containing the specific factor I mentioned before that are able to induce their early osteodifferentiation. As endothelial cellular model, we chose this cell line, HMEC, that basically are microvascular endothelial cells from derma, so basically dermal capillaries. And in order to study the crosstalk between the two, we carry out this two-dimensional co-culture system that are based on the use of transwell insert, the one that you see there, that are characterized by pore with a small site diameter. These pore have a diameter of about 0.5 micrometer, 0.4 micrometer, that not allow, let's say, the passage of the cells from the upper chamber to the lower chamber. So the cell line that we plate within the insert is confined to this compartment. At the same time, soluble factor can easily pass from one chamber to the other, allowing that crosstalk between the two cellular lines. More specifically, we set up two different configurations. First of all, in order to evaluate the impact of endothelial cells on osteodifferentiating stem cells, we plated our mesenchymal stem cells on the bottom and endothelial cells within the transwell. And what we look at was basically the expression of osteodifferentiating marker by mesenchymal stem cells, bone production, the expression of pro-angiogenic factors, and finally, we analyzed their metabolic profile. Contemporarily, we studied the opposite crosstalk. I mean, the impact that osteodifferentiating stem cells could have on endothelial cells feature. Basically, we studied endothelial cell proliferation, migration, chemotaxis, and tubulogenesis. Let's start from the first configuration, in which we have mesenchymal stem cells on the bottom and endothelial cells within the transwell. So we are looking at what change about mesenchymal stem cells. And what we found is that by performing real-time qPCR on mesenchymal stem cells, both shed and ASC, we found that in the presence of endothelial cells, as you can see from this graph, we have a significant upregulation of the genes that are associated with osteodifferentiation, like the bone morphogenetic protein 2 and osteopontin, meaning that the presence of endothelial cells promote the osteodifferentiation of mesenchymal stem cells. Then, in order to investigate the potential ability of our mesenchymal stem cells to provide bone formation in vitro, we use this 3D model. Basically, we seeded our cells, ASC, in the presence or not of endothelial cells within a scaffold, and we kept the scaffold within this perfused bioreactor in order to better control the environment, the physical properties of the environment, and to better mimic the physiological environment, let's say. And what we found was that after 1 month of co-culture within the bioreactor, in the presence of osteodifferentiating stem cells, as you can see from this black column, the amount of newly formed trabeculae is significantly higher, thus demonstrating that endothelial cells are able to increase the amount of mineralized matrix produced by our stem cells. Then, coming back to the coupling between osteogenesis and angiogenesis, what we check was the expression of pro-angiogenic factor in mesenchymal stem cells, culture alone or in the presence of HMAC. And as you can see from this real-time PCR quantification, we found that, of course, osteodifferentiating stem cells shown an upregulation of these pro-angiogenic factors in comparison with the undifferentiating stem cells. But this upregulation is even more pronounced in the presence of endothelial cells, thus supporting the idea that the presence of endothelial cells further enhance the recruitment by mesenchymal stem cells of new endothelial cells by secreting this factor like VEGF and angiopoietin-1. Also, in this case, we observe the same with SHED and with ASC. Finally, we look at the metabolic profile of our mesenchymal stem cells because it is known that osteodifferentiation require an oxidative metabolic switch, let's say. And indeed, through the quantification of the activity of different enzymes that are involved or into the glycolytic pathway, this graph, or enzymes that are involved in the oxidative pathway, in this in the bottom, we found that, as attended, if we compare osteodifferentiating stem cells, light blue column, with undifferentiated stem cells, we observe an increase in their oxidative metabolism and a decrease in their glycolytic rate. When we put our mesenchymal stem cells together with endothelial cells, what happened is that endothelial cells are able to improve the anaerobic glycolysis of these cells, but without affecting this oxidative metabolic switch that is required for their osteodifferentiation. Moving to the second configuration, so aimed at study the impact of osteodifferentiating stem cells on endothelial cells, what we did was to, first of all, look at the proliferation of our endothelial cells co-culture with mesenchymal stem cells. And what we found by performing some cell viability assay was that in the presence of mesenchymal stem cells, the viability of endothelial cells is increased. Similarly, also their proliferation, also their migration is improved. Indeed, as you can see from this assay, this migration assay that is the wound healing assay, basically we plated our endothelial cells in order to obtain a confluent monolayer. Then we apply the wound within the monolayer and we measure the area of this gap over time. Why? In order to assess the migratory ability of endothelial cells to migrate and close the gap. And what we found was that osteodifferentiating stem cells significantly improve the migration of our endothelial cells, so promote the migration of endothelial cells in vitro. This is another migration assay, peculiar migration assay called chemotaxis assay. Basically, again, we use some transwell insert, but in this case, the diameter of the pore is larger. The pore have a diameter of about 0.8 micrometer. Why? Because in this case, the passage of the cells from the upper chamber to the lower chamber is allowed. In this configuration, we can study the chemoattractive potential of mesenchymal stem cells on endothelial cells. And what we found is, again, that in the presence of mesenchymal stem cells, and in particular osteodifferentiating stem cells, the chemotaxis of HMAX, so it's their tendency to migrate towards the lower chamber, is more pronounced, both in the case of ASC, both in the case of SHED. The last assay I would like to talk to you about is the tubulogenesis assay. Basically, in this case, our endothelial cells are seeded on a matrigel. Why? In order to induce, as you can see from this movie, if I can, okay, in order to stimulate the formation of this vascular network. This assay is very interesting for us because it provides a morphogenic readout in a quite simple and reproducible manner. But as our colleague from the Politecnico of Turin will explain you later, this readout is quite easily modelable. Why? Because tubulogenesis assay represents a quantitative multi-parametric system, as you can see from this slide, by simply apply a free plugin, for instance, the angiogenesis analyzer plugin for ImageJ, we are able to measure a lot of parameters about our vascular network, thus evaluating the complexity, the maturity of the network, and so the ability of our endothelial cells to form a more or less complex vascular network. What we observe in this case is, again, that some parameters that are directly related to the complexity of the network, for instance, the number of nodes of junction and master junction and segment, significantly increase when endothelial cells are co-cultured with mesenchymal stem cells osteodifferentiating. By contrast, other parameters that are not directly but indirectly correlated with the complexity of the vascular network significantly decrease. For example, the number of isolated segments. So we found that osteodifferentiating stem cells promote vessel recruitment in vitro, both in the case of shed, and the same was also observed when we co-culture endothelial cells with ASC. You can see that the number of node of junctions, so the complexity, the maturity of the network is higher, and other parameters like the number of isolated segments is significantly reduced. Now, summing up, what we demonstrated was that on one hand, endothelial cells promote osteogenesis by enhancing the expression of some factors that are involved in the osteodifferentiation of stem cells, and contemporarily promote the expression of pro-angiogenic factor that in a virtuous loop stimulate the recruitment of endothelial cells, and so their proliferation, their migration, and their vascular network formation. Now we are trying to deepen this complex crosstalk from a, let's say, omic point of view. I mean that in order to have a broader landscape on what happened during this co-culture, and so better details on the crosstalk between the two, we prepare some RNA sample from the same configuration I showed you before. And we sent this RNA sample to a company to perform a bulk RNA sequencing that now the bioinformaticians of our unit are analyzing in terms of differential gene expression and functional enrichment analysis in order to better elucidate what happened from at the transcriptional level on these two types of cell type when are co-cultured with the other cell type. And if you have any doubt, curiosity, or question, I'll be pleased to answer you. In the meantime, I would like to thank you all for your attention and for my problem with the screen before.
Thank you, Giorgia. Thank you so much. Is there any question from the audience?
Yes, I wanted to ask something. Like your phenotype, let's say, you basically you claim that cells kind of enhance the expression of pro-angiogenic factors or promote mineralization in the crosstalk between between mesenchymal stem cells and the endothelial cells, which then you actually see in the vasculature formation on the hydrogel, though, on the matrigel, sorry. So you see that they kind of proliferate once there on the matrigel, but on the other hand, on the multi-well, when you on the transwell, when you culture them on the transwell, you assess the phenotype by real-time PCR only. So therefore, you know that the expression is higher. But what about the phenotype, the protein expression or whatever? Do you use also some protein expression tests or functional tests or whatsoever? Or do you plan to use that, or do you think that it's unnecessary just to have a clearer idea on how they are influenced within each other?
Sorry, maybe I didn't explain it very well before, but this we used, this configuration for culturing the cells, but then all the functional assay that I show you are made by detect on the cell type that we seeding in the lower chamber by detaching them and use them to do, yes. So all the functional data refer
so on the matrigel, you mean? Yes. They are the same cells?
On the matrigel, in this case, the cells are not plated into matrigel, it's just plated into the well. But,
and then you move to...
And on these cells, we quantify the expression of some gene by real-time PCR. We perform the cell viability assay as well as migration and tubulogenesis assay.
Okay, cool. Yeah, so you use basically the same cells.
I explained it very well before.
I didn't understand that bit, otherwise there was no connection. Okay.
Yes.
Thank you very much.
Okay.
Well, thank you. And thank you, Giorgia, for introducing the biological intricacies. We are trying to propose solutions for today and for providing the presentation of the biological solutions. So I will now share my screen again to introduce the next speaker. These banners, okay. Professor Gionata Fragomeni from Università Magna Grecia di Catanzaro, who will follow through with the presentation of dynamic models and bioreactor design for bone regeneration.
Okay, thank you, Roberta. I try to share my my screen.
Yes.
Okay, you see my screen?
Yes,
we do.
Yes.
Okay. I talk about the dynamic models and the design for bone regeneration. I have a brief introduction, and I describe the mathematical modeling, what is a bioreactors, very shortly, and I explain the examples and give the conclusion of my work. It's a very, very rapid presentation, and I go. Okay. We have seen that the clinical management of bone fractures has become a serious burden on healthcare providers, and in the future, in the near future, will be even more serious due to the aging of the population. The social costs are estimated to be very, very high. You can see on the left of this slide, and every year these costs increase a lot. And for this reason, optimizing bioreactor design and might guarantee the and the supply of physiological nutrients for the cell to the cells, and control of environment to ensure that the cell survival and to promote their proliferation as in a natural bone. And for this we use the bioreactor, but I talk about the bioreactor in the next slide. What is bone tissue engineering? Georgia explained this very well. I give only a rapid view of this, and the tissue engineering may help overcome limitation of the graft for treatment of large-size bone defects. The determinants of the tissue engineering constructs are the donor cell, the biomaterials, and the stimuli, the bioactive stimuli done to the cells. Georgia explained also that the bone tissue is a very complex tissue. Every tissue have a different behavior. Bone cells have high oxygen consumption, and this consumption is very high compared to the other tissue. The transport occurs through complex in vivo vascular system, the Aversian and the Volkmann systems. But what is a bioreactor? A bioreactor is in this slide you can see different type of bioreactors. A bioreactor is we can define a bioreactor as a device that provides the transport system of the nutrients to the cells, and allow the different removal of toxic and the products of the cellular metabolism. The requirement for a bioreactor are the generation of the flow patterns of the culture and medium. For an efficient removal of toxic products of cellular metabolism. The requirement are these, and also the increase in the in the transport of nutrients from the culture medium to the engineered constructs, and the physical simulation of the constructs during the development of the construct, and also the online control of the chemical and physical parameters of the culture. I talk very rapid on the bidimensional versus three-dimensional cultures. The advantage of bidimensional cultures are associated with simple and low-cost maintenance of the cells culture. And with the performance of functional tests. But unfortunately, the cell adherent cultures bidimensional cultures have numerous very big disadvantage. First of all, the bidimensional cultured cells have not mimic the natural structure of the tissue. In this culture method, cell and cell extracellular environment interaction are not representative. These interaction are responsible for cell differentiation and proliferation, for the vitality and for the expression of the genes and proteins. And this is not good represented. In the bidimensional cultures are not representative of the real cell environment because the growing on a flat surface is not a good way to understand how cells grow, and in a human environment, in a human environment, yes. There is also not a new cell grow, cell grow in a standard bidimensional culture. They consume growth media and exude waste. This can result very dangerous for the other cells, and this generate waste products like dead cells or nutrition waste, and damage. This can damage all the cells, all the environment. Another big factor is the static versus the dynamic culture method. The static cell culture is the method of growing the cells in a fixed and stable condition, such as stationary dish or a well. Dynamic cell culture is the method of growing cell in a changing variable condition, such as rotating vessels. In this slide, you can see different types of bioreactors. On the left, there is hollow fiber bioreactors. On the right, there is radial flow bioreactors. For the hollow fiber, you can see that there is an analogy of the hollow fiber bioreactors with the tissue and the organs. There is an analogy between the method of the membranes that are like these, that are like a vessel, a vascular bed, and the cells surround these vessels, this membrane, is like a natural condition. But these are very simple to design and to test, but have very high limitation. We modeling mathematically these bioreactors, and the models, the mathematical models are very helpful both in the design phase and of the bioreactor and in the stage of the tissue maturation under the tissue engineering, typical in tissue engineering condition. These are the phases that we are following. We start from domain definition. We define the boundary condition. We set also the physical property, the parameter definition, the initial conditions, and then we have the iterative process, and we have the results. We use the software COMSOL Multiphysics, that is a software that use the finite element method, and is a commercial software. Why we use? We have simulated both hollow fiber and radial bioreactors. Why did the radial bioreactor are better than the hollow fiber? The perfusion of this kind of bioreactor is in the medium culture is radially, occurs radially, and towards to the cells, and these For this reason, the characteristics distance of the solutes and the cells are smaller, while larger passage surface are available, and this allows the tissue to be developed using low surface velocity, low velocity and pressure. This is very only a slide to describe that very well described Georgia, and for explicate the role of the vascular system. As Georgia said, the vascular system, the bone are very, the vascular system in the bone are very complex and very difficult to reproduce. We use some scaffolds. Very important are some parameters like the porosity, the pore size, the interconnectivity between the pore and the surface area. This is a radial bioreactors, and in this zone we have the medium, the scaffold. In this zone we have a central channel, and in the left or in the right we have the annular vessel, the annular channel. We use the Navier-Stokes equation in order to describe the void space, and the Brinkman law in order to describe the flow inside the scaffold. We use also a dimensionless approach in order to have a more simply solution to analyze. These are the parameters that we have studied for this bioreactor, and the boundary condition. This is a symmetric bioreactor, and then we use the symmetric condition. The axis, the axis is impervious to fluid. There is a fully developed inlet profile, and also for the outflow. And there is a continuity of pressure and velocity at the interface, and there is we impose a no-slip condition in the non-adherent condition in the walls. as you can see, there is a mesh very, very small in the interface zone. This is this zone, this small zone, and we optimize this mesh in order to have a minimum error. And we simulate, for example, the velocity for different Reynolds number in input, and we have this is the scaffold, this is the inner cavity, and this is the outer cavity, the outer channels, and we have the results for different number of Reynolds. This is the small round number of Reynolds. This is the medium, and this is the maximum number of Reynolds. And we have also simulated asymmetric packet bed bioreactor and asymmetric bioreactors, and we have an inlet. This is a scheme of the bioreactor. We have an inlet at the center, and we have also an outlet on the lateral side. This is a prototype of the bioreactor, and this is the scaffold that we used for the experimental analysis. We use also for this an adimensional approach in order to have a more efficient, more simply solution. And also in this case, we have seen the scaffold that is in this zone, and the void zone that is this and this. And we have also for this the Navier-Stokes equation in the void space, and the Brinkman equation in the scaffold zone, in this zone. We also simulated, we have also simulated the different conditions, for example, different number of Reynolds, or different position of the outlet, and these are the boundary conditions. We suppose that we have a parabolic velocity in the inlet in this zone, and also other type of boundary condition, and we simulate these are the results, and you can see that the outlet give a distortion of the velocity in this zone. This is the concentration at different inlet condition at different number of Reynolds. You can see that for the low number of Reynolds on your left, we have that there is a shunt in the upper zone of the bioreactor. For the central case, we have a radial flux distribution that is uniform, and in the In the last case, in the higher number of Reynolds, we have a value concentration that oxygen concentration that is very low in this zone, in the central zone of the scaffold. We have plotted also the this kind of result. On the left, you can see the effects of number of Reynolds on the model predicted by reactor and on the fluid dynamics of the bioreactors. And on the right, you can see the tracement, the tracer simulation for compare these results with experimental results. This is the shear stress. We have also say it in different time that mechanical stimuli can influence the behavior of the cells, and in this slide you can see that different number of Reynolds, different inlet condition can influence in different mode the shear stress, then the mechanical stimuli. In conclusion, the mathematical models of bioreactors is a good help both in the design phase that we can see that are the results in for the bioreactor and also in the working phase of the bioreactor in the typical condition of the tissue engineering. In fact, they represent a useful tool both in the optimizing the design of the scaffold, as well as the outlet selection and the section and the position of the outlet. And in case of the bioreactor is existing, is realized, we can prevent the mass distribution of the radial flow by optimizing the inlet flow rate to the bioreactor. Okay, thank you for the attention.
Thank you, Gionata. Thank you so much. Are there any questions from the audience? Okay. So thank you again, Gionata. And
thank you.
We now move to the next presentation. And actually, we keep talking about mathematical models and the computational models. I don't know if you can see my screen. Yes, okay. Because the next presentation will be given by Professor Tommaso Lorenzi and Dr. Nadia Loi from the Department of Mathematical Engineering of Politecnico di Torino, who will introduce mathematical modeling approaches to abstract away from the biological complexity involved in this kind of in vitro models of bone remodeling. And then we will move with Riccardo Smeriglio from the Department of Control and Computer Engineering, always from Politecnico di Torino, talking about the implementation of said mathematical models in complex computational frameworks for the simulation and optimization of in vitro model designs. So I will leave the stage to Tommaso and Nadia for starts, and then Riccardo will continue their presentation.
Thank you, Roberta. Just a second that we share our screen. Do you see our slides?
Yes, but not full screen.
Now, in a second, it should be.
Yes, yes.
Are they full screen?
Perfect. Thank you.
Okay, thank you so much. So as Roberta mentioned, we are going to give let's say, short but broad talk, focusing more on the mathematical tools rather than on specific applications. And these are tools that we have been using in the past few months, also in collaboration with Roberta and Riccardo, to tackle biological problems which are linked to those that Giorgia presented in the first part of these workshop talks. We have been doing so, Nadia and I, in collaboration also with Dr. Fiona McFarlane from Certara Inch. She was previously at the University of St. Andrews in Scotland. And the type of tools that we have been using are mathematical tools that can be employed to describe different aspects of cell dynamics, being them spatial evolution or spatial and evolutionary dynamics. In cellular system, let's say in a broad sense. So we will be talking about cell populations, and then this can be interpreted in different ways depending on the specific biological problems that one is then interested in tackling. The type of research that we carry out touches upon different aspects of mathematical modeling. So on the one hand, we are interested in the formulation of individual-based models, that is, models which track the dynamics of single cells. As I said at the beginning of this presentation, both from the point of view of spatial and evolutionary dynamics and from the point of view also of integrated spatial and evolutionary dynamics. We are also interested in how, starting from this type of models, we can recover through coarse graining procedures, continuum models, that is, models that describe the aggregate dynamics of the cells as a population and not at the single level, or to formulate straight away continuum models for the dynamics of cell populations without departing from underlying individual-based models. And then typically individual-based models are very accurate but quite complex to be studied from a mathematical point of view. And so when we have an individual-based model, we typically rely on numerical simulations. Whereas then when we know the counterpart, the continuum level of this model, which is usually formulated as a partial differential equations or a differential equation in general, then we can combine together mathematical analysis and numerical simulations to address different type of biological questions. And our expertise so far have been mainly applied to the mathematical modeling of cell movement, tissue development, and tumor growth. And then we have been transitioning into considering also processes like bone remodeling and the formation of networks of endothelial cells, again, mainly in collaboration with Roberta and Riccardo. The methods from a mathematical point of view that underlie this type of research are borrowed from the theory of random walks and velocity jump processes, reaction advection diffusion equations, kinetic equations, and structural population equations, that is, equations which describe the dynamics of populations which are structured with respect to some physiological traits, such as the level of expression of some genes, which can then be linked to the phenotype of the cells. And a first type of individual-based models which can be considered in this framework are on-lattice individual-based models, which means essentially that we look at our cell population as a collection of agents which are distributed on a lattice, which represent a discretization of the physical space. And then the movement and proliferation and death of cells is described as a branching random walk. That is, the single cells perform a random walk in the physical space since they move. And it's a branching random walk in the sense that they can also divide or die during their dynamics. And so typically in this modeling framework, one first of all defines a set of rules which can model this type of phenomena. And like in the schematic given here, you can imagine to have rules which describe the probability of cell proliferation, the probability of cell division, and the probability for a cell to remain quiescent. And then you can also incorporate into the model, this is for a one-dimensional case, but it could be extended to 2D or 3D with minor changes. You can take into account the fact that the cells also can move in the physical space by jumping over the spatial lattice. And then, as I was mentioning at the beginning of the presentation, starting from these individual-based models, using suitable limiting procedures that I'm not going to discuss today, one can derive a corresponding, let's say, continuum model that describes the aggregate dynamics of the cells. So the previous class of models describes the dynamics of single cells, and so in a sense provide a microscopic level description of cell dynamics, whereas the type of models that we consider here upon this upscaling describe the macroscopic level dynamics of the cells in the sense that we look at the spatial dynamics of the cell population. And as I was saying, the mathematical object that defines this type of models at the continuum level are partial differential equations of the type of the one which is given here, whereby we have a function that describes the density of cells at a certain position at a certain time inside the system, and the equation captures the effect of cell movement, which can be a form of diffusion, or it can be a form of chemotaxis or apoptosis or directed movement more in general. And then we have terms which capture cell division and death. In this type of underlying individual-based description, we model cell movement as a collection of jumps along the lattice that correspond to the physical space. And this is a sort of strong simplification, and we can overcome this simplification by considering another class of individual-based models, which are off-lattice individual-based models.
Yeah, so one can also consider microscopic models, so individual-based models that are in which cells are not located on the lattice, but cells can be identified simply to their position and to their velocity. And essentially, they are assumed to perform a free drift and a run and tumble. So we also have this velocity of a cell that can evolve because of the run and tumble, so because of instantaneous reorientations. And the run and tumble is typically described as a velocity jump process. So from mathematically, what is done, so we consider discrete in time stochastic processes for both the position and the velocity. So in a time step that we call delta t, one can have a stochastic, so a process that's deterministic for the position, so that describes the free particle drift along the velocity v. And then we have an actual stochastic process for the velocity that at time t plus delta t may change because of external forces. So, for example, we can have chemoattractants that may be included in the external force. And then we may have this velocity jump process that describes the run and tumble. So this is described by means of a sigma, that is a Bernoulli random variable that has a parameter delta t. So if this random variable is 0, then we have no tumble, while if it is equal to 1, then we have a velocity jump. And the velocity changes into a new one that we call Bt prime, that is distributed according to a certain distribution that can be measured. So for example, if we assume that this run and tumble, these microscopic reorientations of cells is the contact guidance along the fibers of the extracellular matrix, then one can measure the orientations of the fibers and can choose this distribution Q as the distribution of the fibers. Then for these kind of microscopic models, there is a first level of continuum limits that are represented by kinetic equations. So for this stochastic process given by the position and the velocity, so we can formulate kinetic equations on the phase space xv, and this gives a statistical description of the dynamics. So we don't need to perform a microscopic simulation of the microscopic model in which we need to choose the number of cells, but here we have the full statistical portrait of the dynamics that is described. So essentially, we don't consider n cells, but we have a distribution, a probabilistic distribution function f that depends on time, position, x, and velocity. And can derive formally an evolution equation for this distribution function f, in which we have the free drift, so the role of the external force, and then also the velocity jump. From this equation, we can derive macroscopic models, so in the same sense that Tommaso explained before. So we can introduce a macroscopic density ρ, so the cell density at position x at time t, and also their average velocity, and this can be done by introducing the statistical moments of f. And then in appropriate regimes, by using appropriate techniques, one can derive different macroscopic models. So for example, if the regime is a diffusive one, then one can derive a model that is similar to the one that Tommaso showed before. Or if the parameters dictate a more drift-dominated phenomenon, then we can have another kind of macroscopic model. And here, all these quantities that appear, so the U, the D, so the quantities that define the macroscopic model, actually come from the details of the microscopic model. So the details of the microscopic model are inherited by the macroscopic one.
And then just to conclude, we wanted to mention the fact that this type of modeling approaches could be extended in order to capture the presence of phenotypic or genotypic heterogeneity, which are typical inside cell populations, because so far the models that we have presented, they describe only the spatiotemporal dynamics of the cells, but one might be interested also in incorporating into the model the effect of evolutionary aspects linked to the fact, for instance, that depending on the environment in which the cells are embedded, they can undergo phenotypic changes. And so, in essence, what one can do can introduce in this type of models additional structuring variables which describe the dynamics, which describe, the cell phenotypic states, and then using appropriate extensions of the limiting procedures that we typically use in order to derive the mesoscopic and macroscopic models in the setup of the models that we have presented so far. One can recover again a microscopic model which captures not only the spatial and temporal dynamics of the cells, but also the way in which their characteristics, i.e. their phenotype, can evolve over time. And this is something that, again, can be done for all the models that have been presented so far. And then we have been starting collaborating with Roberta and Riccardo in particular in order to try to develop models which can be biologically relevant for the problems that were introduced at the beginning of the workshop or the presentation today. And we now leave the stage to Riccardo, who will present in more details the computational models which can be linked to this type of mathematical approaches.
Thank you so much, Tommaso and Nadia. And Riccardo can start with his part of the presentation.
Or in the meanwhile, may I ask a curiosity, please, Roberta? May I ask?
Sure.
Yeah, of course.
Thank you for your very nice presentation. And I apologize for my question. I'm an experimental biologist, but my curiosity is, is your space in your simulation homogeneous, or can you introduce changes in homogeneity? So these homogeneities in your space, is it possible in your system, or in lattice or off lattice systems?
Yeah, in both. Yeah,
in both approaches.
Yeah, the environment may be heterogeneous, so it may depend on space, and it may be different from position to position. Yes.
Okay, so you can design the shape of inhomogeneity.
Yeah.
So that's my question. Okay, that's very fine, because in some systems, but we can of course we can discuss later on. But in some biological systems, different kinds of inhomogeneities in the space, in the 3D space, could occur, and so that's very important for us to account for that. So that's a good.
Thank you for the question. That was not naive at all, and that will certainly lead to possible avenue for collaboration. So thanks.
Okay, thank you. Thank you.
Roberta, may I ask a question as well?
Sure.
I'm Gerardo Catapano anyway. Thank you for your very nice presentation, quite intriguing. I was wondering, you were showing that you accounted for the effect of a force acting on the cells according to a linear model. You split the effect of the force from the own movement, mobility of the cell, if I remember correctly.
Yes.
Now, I was wondering, how close is that to reality? Because let's assume that the cell is subjected to a flow field. I may expect that the action of shear stress may also influence the drift as well as the run and tumble behavior of the cell. Is that correct, or am I?
Yes. Yeah, that's exactly. So, for example, here you may have the run and tumble. So if you want the movement, the random movement that the cell a priori performs, but then, for example, if you have a drift, so for example, I don't know if the cell is in a fluid that is running. So if you have a drag force, this is included here in the external force term. So of course, it depends on what you model in which terms. So this is more for the run and tumble, and this is for what you can consider the external force. There are also people who consider chemotaxis an external force, other people who merely model chemotaxis in the run and tumble. That's debated also from the mathematical point of view, but yes, that's what you are saying before. So you may have here a drift and here the run and tumble.
Actually, I was not thinking of chemoattractive forces. I was thinking of a sheer mechanical force. I mean,
also here, here, for example.
No, we have done experiments, and we have seen that if you superimpose certain shear stress to a mixture of cells containing endothelial cells, the endothelial cells tend to align with the flowing fluid. And I don't know how you can consider that in this model, or probably do you think you have to change the way you model the actual movement of the cells, so you can still simulate this kind of action on the cells with this model, with this linear model?
Yes, I would include the shear stress, so the force you were mentioning, in this term. So this is linear only with respect to time, but the force may depend on both the position and the velocity, both in a linear way, but that's not compulsory. I mean, the external force may depend on the velocity in a nonlinear way. That's possible.
Okay, I was thinking of the effect of the force on the other two terms. That is to say, if you have that cells change their shape and they adhere more strongly to the surface, the shear stress is going to change also the way, or the rate at which cells drift or run and tumble. This way. Okay, but that's probably in the next talk. Riccardo Smeriglio is going to tell us more about it. Thank you anyway. It was very nice.
Thank you. Thank you.
Oh, good afternoon, everybody. I'm going to share the screen right now. So let me do it. Okay. You should be seeing my screen. Okay, can you see my screen? Okay, perfect. So I'm here to talk about multi-model and multi-level simulators for, in particular, optimization via simulation. So as Giorgia and Gionata described very well, living systems and biological systems are very complex. So they can be modeled, as Tommaso and Nadia were describing, using mathematical models that are quite abstract. So, of course, the high complexity of the biological systems will drive the high complexity also of the model, as we have seen. So this will always be a problem, we can say. The complexity of the model will be always high. So the actual challenge in this context for us is to use in silico modeling, so to model these in vitro processes that we were talking about in silico, and they try to optimize the actual protocol to get a goal. So the main idea is to try to use to optimize the protocol and the stimuli that we want to give at the in vitro model in silico, and then move the same information on the actual in vitro model. In particular, for this kind of things, the best suited models are multi-level model and hybrid models, or multi-level hybrid models that merge both the multi-level stuff and the hybrid capabilities. In particular, multi-level models basically represent at least two organizational levels. This is highly important in the context of biology because also biology, a biological model like a biological system will merge a lot of different organizational levels, from organ networks to cellular internetworks and molecular networks and so on. So this is very important also for the computational model to mimic very well the biological model. In the same context, a hybrid model can combine different formalism. So from the point of view of the modeling, having different formalism help us also to get more capability to merge different processes together and to interface everything that can be interfaced with this kind of formalism. So in particular, these multi-level hybrid models are good because they can give us richer detail, of course, incorporating different structures, but also better accuracy because they mimic better the biological system. In particular, one good example of a multi-level in silico model, hybrid in silico model, is a PhysiBOS 2.0, that is the simulator that we are using right now, and is actually composed of three different frameworks that are PhysiCell, BioFVM, and MABOSS. In particular, they basically are. PhysiCell is a multicellular simulation framework that allows to simulate the actual cells and their behavior, so their movement, as we were describing inside a microenvironment, but also their mechanics, cell growth, and all of secretion things that we can imagine. In the meantime, they will move inside an environment that can be 2D or 3D, and this will be done by BioFVM, that is a solver that will solve a partial differential equation to mimic the diffusion of all the substances. For example, as you can see in the image, we can deal with oxygens or every kind of stuff, so also drug or specific activator or inhibitor. In the end, there is MABOSS, that is a continuous time Markovian simulator. Here you can see the hybridness of this model because we are using two different formalism, the partial differential equation, and also Boolean model used by MABOSS. And this one will simulate the Boolean models to mimic the regulation and the intracellular signaling networks. So the main idea of PhysiBOS is to merge all of these three interesting things in order to have something that is able to actually simulate a full biological system and its behavior. So it is very important to be able to simulate this through time and space. Of course, it has different capabilities. For example, it has the capability to simulate special 3D simulation, of course, to So cell behavior, cell fate, but also differentiation. It's really interesting that we can also include different phenotypes. For example, as you can see in the figure at the bottom right, we can include also different type of cells, and we can see the transition from epithelia, so the differentiation from epithelial cells to mesenchymal cells. And this can be really interesting in the context of bone remodeling. So it actually allows to simulate most of the biological system, actually getting the hands dirty, we can say, and actually using its capabilities. So our main idea is the one to use these modeling frameworks to actually in silico to model in silico the biological system, and then to try the different protocols, in particular by optimizing these protocols but in silico using computational models, because of course optimizing protocols in vitro is very difficult, because it's of course computation require a lot of time and a lot of resources, because each trial, like in a normal trial and error optimization method, we would have the problem that if the actual protocol that we select is not the correct one, and we are getting something that is basically wrong, we have to basically throw out everything and restart from the beginning. Moreover, a lot of times these kind of protocols are derived by previous knowledge, but are not systematically optimized. But of course, optimizing something systematically in vitro is very difficult. So the main idea is the one to, you can see in the figure, to model the biological system, for example, a bone, and in the context of the bone remodeling. Then once we have this in silico model, we can of course run several simulations of this in silico model, and we can merge this simulator with artificial intelligence optimization methods that allow us to optimize systematically the protocol and the stimuli that we want to give to the specific system. Not only the protocol, but also all the parameters of the substances and whatever we have seen. So this main idea is method has finished, and move it on the in vitro trial. So the idea is actually to develop this protocol in silico and then move on to the in vitro part to try and see if this protocol is actually good, but at least we have a starting point for making different protocols. So we have a starting point from a protocol that is working very well with a computational method of the specific biological system. That's the big idea. Of course, optimization methods, there are a lot of simulation required. In particular, we are talking about thousands of simulation. But even if these biological, like these computational methods and simulator are quite fast compared to the in vitro cell growth, of course, they require a lot of time. We are talking about 30 seconds of a very small simulation. I know that for a single simulation, this is not that much time, but if we want to run thousands of simulations, this is still a bottleneck. So if the bottleneck, if the computational complexity and the simulation time required by this kind of simulator, one of the most important solutions is to use surrogate models. Surrogate models are usually artificial intelligence models that basically will replace, we can say, the simulator and mimic the actual capabilities of the simulator by simulating this model. By running this model, they will be able to provide the same answer of the simulator, but of course they will not be white box, but they will be actually black box. So we lose the information about the model that is, of course, inside the actual computational model, but we gain a lot of speed, we can say. So as you can see in the figure at the right, for example, 70 simulation will require more than 1,000 seconds. And this is too much if we want to run 70,000 simulation. So in particular, we developed in a quite easy use case, in particular our tumor spheroid subjected to TNF stimulation. We developed a LSTM, long short-term memory network, surrogate model to replace the simulator. In particular, the idea is to use this long short-term memory network, but at least recurrent networks, because they can allow us to consider the time. Okay, so it's very important because all the biological systems are evolving through time, so we don't have to do like a screenshot of the actual step, but we want to see its behavior and how it is evolving through time. So it is important to create and use models that are capable of dealing with time series. In this context, we developed this long short-term memory network, and as you can see in the figure at the left, it was actually able to reproduce the simulation in a quite good way. The dashed line represents the predicted simulation from our new model, the LSTM, while the continuous line represents the original simulation got from Physibos 2.0. In the meantime, as you can see in the figure at the right, it can actually reduce the time. As you can see, there is still the training time that is important and is of course impacting, because this kind of models needs to be trained. But once the model is trained, the inference is quite instantaneous, so it can replace the simulator by giving us the output of the simulation and also how the cells are evolving through time, basically instantaneously. And in this context, running thousand simulation is becoming very well, because in these optimization methods that we want to implement, the design space exploration, so we have several solutions for each protocol, and we want to explore the space of this solution. So we want to run a lot of simulation to explore in a good way all these this design space. In conclusion, of course, these multi-level and hybrid models have the potential to infer the behavior of biological systems, of course, reducing the time, but this reduction is not enough to run the optimization via simulation. So the idea is to run this optimization via simulation that can accelerate the systematize the development of this protocol, but using this surrogate model to replace somehow the computational model. In the future, we would like to explore the capabilities of artificial intelligence to generate things, as we are looking now. They are like very important right now, the generative model. So we want to try including some generative model, not to actually start from a pre-existent protocol and then trying to optimize it, but actually generating some protocol using these capabilities. Thank you all for your attention.
I can see there's a question from the audience, Konstantinos Theophilatos.
Yes, hi. Congratulations for this presentation, very nice. I have a question regarding the training of this Physibos-based model. What is the minimum requirement of samples you need to have to do this efficiently?
We actually trained that model with, if I'm not wrong, 5,000 simulations, and it was actually able to reproduce the behavior of the simulator. Of course, the number of needed experiments, we can say, to train it increase a lot when we want to simulate some more complex, we can say, system. So if we want to do it with a bone remodeling system that is quite complex, as everybody has described before, we have to train it with probably more simulation. But the number, I think it depends a lot on the specific system that you want to mimic, and also on how much the different protocols will vary the output. Because if the outcome is basically the same varying the protocols, it's quite useless to train a computational model doing this. If even a small change in this kind of inputs can actually change a lot the output, the outcome, so that will require more samples, but for sure 1,000.
And in addition to the number of cells and its status that you provide as input, which are the other input characteristics that you feed into the model?
Okay, in this context, that specific model was done for this system of this cancer spheroid, and it was taking just 3 parameters that were the TNF concentration, the TNF, so tumor necrosis factor concentration, and the tumor necrosis factor pulse period and duration of each, we can say, administration. So basically, the model was actually trained using the output of the simulator at each time step. We decided to use a time step of 30 minutes. So for each 30 minutes, we were giving him the number of alive, apoptotic, and necrotic cells as a training point, and also, of course, the input. And the backpropagation was done with all of these outputs. So basically, the network, once it's done, it's trained, take as input the protocol, so TNF stimulation and what I said before, so the 3 parameters, and then gives as an output the number of cells at each time step. Of course, this means that also this is a problem of long short-term memory network. The error is propagating through time because, of course, the output of the first step will be used from the specific network, we can say, that needs to produce the output of the second time step. So the first time step will be given as an input to the second one. So if there is an error in the first one, there will be a bigger error in the second one, and that's a problem that we have to address, but it's still a problem inside the long short-term memory network. That's why it needs to be trained very well.
Thank you very much.
You're welcome.
I see a question is coming from Professor Munaron. Please go ahead.
Yes, thank you, Roberta. Thank you for a very nice presentation. I think that's a good, but may I ask you if your opinion is that moving towards high throughput experimental approaches would be a good solution for you, or not? Because the point is that high throughput experiments, of course, provides a lot of data, a lot of experiments, and so that's good. But the control, the experimental control is less accurate compared to low throughput experiments. So what is your opinion about it?
My opinion is to use neither one, nor one, neither the other. Okay, so basically the idea is when you use an optimization method, the first phase is called exploration, usually, where the actual method needs to explore. So some methods will also use some random exploration way. So they explore randomly the design space of exploration, the design space of solution. While in the end, there will be the phase of exploitation where we found, for example, a spot that seems to be promising, and the model is actually going over there to look into it and trying to use what is near to the spot. So the main idea is to use this high throughput model to perform the first phase, so the design, the first exploration phase, where basically we want, yes, to know which is the most promising way, but we don't have the actual needs to very understand in a deeper way actually why it's that way the results. In the phase of exploitation, for sure, but also in the phase of verification of all the results that we have, the most important thing to use is the actual simulator that is not the actual multi-level simulator that is actually simulating it, and it's more easier to understand also from a biological point of view. So I thank you for the question, because we are also concerned about that. But I think that the best way is to use the one that is faster to do a big exploration of the environment, we can say, and then when we want to actually get the best protocol, to use something that is also reliable as a biological point of view.
And I think that a second point, if I may, I think that the second point you raised that is very important for us is that error, that you propagate error. So if I understood well, this means that it's very important to have an experimental readout that is with a very low degree of artifacts. Otherwise, this experimental error should be amplified over your simulations. I understood well.
Yeah, and this also depends a lot from the actual data where we are going to train, because if something is very stochastic, so it will give us very different results from the same, that's a problem of physics, but it's also a problem, let's say, of biology, so that biology is quite stochastic with the same stimulation, not always the outcome is the same. So using this kind of training inputs, of course, the network will be able to provide the network is not stochastic. Once it's trained, when I give him as input, the output will be always the same. This means that, of course, if the output is in the middle of the actual output, that will be wrong. And of course, if during the training phase it was producing an error, and if the first step is already different, this difference will be also propagated through time. That's why, for example, I have not the plot right now, but if we see the actual error inside the simulation, of course, at the last time step, the mean error was higher than at the beginning, because at the beginning it's the same, while we were, as long as the network is producing its output, the error will increase. That's why I don't think we can actually trust this kind of model for the exploitation phase, so where we have to be very precise. But still, let's say that the behavior of the whole model was actually quite well represented. So, for example, just to say something very stupid, we can say, if I give him no stimulation, also the network is able to say that the cells are not dying. Okay, so that's what we want, for example, at the beginning in the phase of exploration. Then in the phase of exploitation, we have to get more information, and for that we will rely on more precise models like this Physibos one, even if they need more time to be executed. Like that's a good time. It's not a waste of time. Run something to be quite sure that the outcome is significant.
Thank you, Riccardo.
Okay, thank you. And as we are approaching the end of the seminar, I will take the time to do my presentation on the interdisciplinary fil rouge, we can say, that is connecting many people who presented today with, which is the Saisei project. I'm trying to share my presentation in presenter mode. Can you see it? Yes. Okay. I will also.
Yes.
Thanks. Okay. So the main point of this final talk is to highlight the connecting dots, let's say, among all of the previous talks, for which I warmly thank all of the speakers. I warmly thank them because I think that from their presentations, something very interesting emerged, which was the diversity of perspectives that people from different scientific and technological backgrounds can have on the same problem, especially if the problem is complex. This reflects a lot, in my opinion, in language. Just think about how many shades of the word model we use today. The simplest acceptation of this is the using model as in vitro model or in silico model. As we are working with colleagues from the same background, a biologist working with a biology will say model to say in vitro model. They don't maybe need to, we don't maybe need to say in vitro. A mathematician with a mathematician will say model to subhuman mathematical model. A computer engineer for a computational model. I'm trivializing, but that's the idea. In this presentation, we use the same word for saying many different things. And this is a good signal that the perspectives that we are gathering together, they can build a shared and interdisciplinary perspective. And this is the main point of my talk today. So the problem at hand for us is the systematic design, computational design of in vitro models. That was mentioned also by Riccardo just now as the computational design of fabrication protocols. Let me connect to that. The engineering of in vitro models for basic research or pharmacological research is, as Giorgia and Gionata detailed very well today, a very complex endeavor. And if we see the same topic from the perspective of fabrication, we can say that the parameters that we use for the design and fabrication of an in vitro model affect the functionality of that model in supporting its scientific aims. For example, the kind of experiments I can perform with my model. For example, if we stick with the microwell, transwell-based co-culture that Giorgia was mentioning, the mere size of the pores can affect which kind of phenomena I'm going to observe in my system. For example, if the pores are less than 0.5 micrometers, cells won't move across the pores. If I want to study chemotaxis, I need larger pores, for example. And our goal with computational optimization via simulation that Riccardo was mentioning is to design intentionally our in vitro models so that all of these features, even the less obvious ones, are controlled in a way that makes the desired properties emerge from the in vitro model. And this is, of course, a call to interdisciplinarity, because I don't have to describe why the entirety of these talks did already. The more perspectives we can gather around the complex problems, the more information we see, and the more information we can use to tackle it. And just to be specific in defining interdisciplinarity, this kind of effort, in my opinion, stays in the passage between multidisciplinarity, so the conditioning where multiple disciplines converge on shared objectives, but they work as a separate endeavors, to interdisciplinarities where the disciplinary boundaries are blurred, and one of the results of the project and the process, besides scientific results, is the creation, the shared development of a integrated knowledge and a shared understanding of the complex problem. And I think that the consortium that held this seminar today is doing exactly that. So moving from multiple perspectives to create a merged one. And in particular, I'm going to introduce the PRIM project that was the occasion for us to join our efforts in this consortium, which is the Saisei project. Briefly speaking, the Saisei project aims to computationally optimize the design of fabrication protocols, biofabrication protocols in general, fabrication of in vitro models for bone remodeling in particular. And the methodologies that it puts into place to do that are multi-scale simulations of the in vitro model, optimization via simulation, which leverages such simulations to explore the design space of the in vitro model and to find some optimal spots for the design, and it relies on the implementation of said in vitro models in the laboratory, both static culture and dynamical cultures, for the in vitro validation of the computationally designed protocols it generates. The expected results for these projects are, first of all, a robust and scalable computational framework for the simulation and optimization of protocols, and the in silico and in vitro validation of the generated protocols. So the high-level objectives coherently are developing multi-scale modeling of the in vitro system we are going to target, and simulating such in vitro models in silico with a multi-level and hybrid strategy, as Riccardo was mentioning, implementing models that are proposed in the first instance as mathematical models, as Tommaso and Nadia were describing. This scalable simulation and optimization framework has the sole, let's say, sole objective of generating an optimal protocol, trying out different configurations in silico to reduce the time and cost required for the trial and error process that would otherwise be carried out in vitro, and go back to the lab with an in silico optimized protocol to test out. This is the idea. And this requires, of course, a validation, iterative validation process, which validates several aspects of the framework and the model. For example, plain model validation, where we try to understand if the information, the knowledge we encoded in the model actually reflects in the real biological behavior, and scaling up of the complexity of the use case that we target with our iterative process, meaning in the beginning we try to test out all this overall framework with a simple biological use case, and then we scale up, adding biological complexity, and see if our approach lives up to the challenge. So in this perspective, we start working with static co-culture, transwell-based co-culture of mesenchymal stem cells and endothelial cells, which Giorgia described in her talk, and let's say the higher in vitro complexity we are targeting is otherwise a dynamic culture, where the cells are also stimulated by dynamically regulated culture conditions, as Gionata was introducing. And what I want to point out from this scheme here is that besides the design of the biological in vitro model, for the overall project perspective, what's important to us is that each one of these differently biologically complex configurations has some tunable parameters. For example, the interface filter pore size in the transwells, the composition of the culture media in the two chambers, the stiffness of the gel we might seed the cells into, and has some parameters, process parameters that can be regulated dynamically. For example, the partial pressure of oxygen in the culture medium and the shear stress that we impose to the cells. Also, we can intend these use cases as something we can extract readouts from. So, given a certain complexity, we can decide to read more or less complex data from our culture. For example, the concentration of some elements in the medium, the temperature, or some more strictly biologically defined properties, such biochemical properties, biological properties such as molecular analysis, gene expression, presence of certain proteomic markers, etc., or morphogenetic behavior of cells that underwent one versus the other condition. This is because our objective is to model and to optimize what we call a biofabrication protocol. So the initial setup and design of the in vitro model and the process parameters we control along the culture. To be more specific about this, the way we see a biofabrication or a fabrication protocol can be outlined by this figure. So we know that we can control some stuff in the process, for example, the initial number of cells, as we said, the interface filter pore size, the initial culture media composition, the properties of the gels we seed the cells into, and this we can ascribe to a fabrication phase for our in vitro model, and then we can control some aspects along the maturation of the construct, for example, the culture conditions in general, the administration of specific biological signals, and the control of fluid mechanical simulation in particular. According to what we measure, we can analyze our culture in time or at the end of the culture to assess how each part of the protocol is affecting the performance of the in vitro model. This is to basically obtain two things at the end of this project. The computational framework, and which is able to simulate and optimize the protocols, and at least one in vitro validated protocol following the scheme I was presenting before. This is contextualized in a long-term vision of speeding up and making more efficient and more effective the process of designing computational not computational, but in vitro models for basic and applied research. This is also in the perspective of moving towards more complex application scenarios, such as regenerative medicine and tissue engineering. This project is carried out by a consortium, which is the one that you are meeting today during this seminar, composed by Politecnico di Torino, and in particular the Smiley's group. Professor Di Carlo is also connected to this call. Riccardo and I are part of this group, and we are coordinating the project with University of Catanzaro and University of Turin, who work at the in vitro validation, developing static and dynamical culture use cases. And part of the Politecnico di Torino unit is Professor Lorenzi and Dr. Loi, who you met just before, that are working on the mathematical modeling aside the computational modeling and optimization. So what the take-home message for this presentation is actually that besides the scientific results that we are obtaining together as a consortium, from the start we said that one of our objectives was to create a shared language with the higher objective to create a shared perspective over a complex project, because we strongly believe that a truly interdisciplinary approach is the only way to really tackle complex projects and problems such as optimizing the fabrication of complex in vitro systems having both biological and technological complexity. So this ends my presentation. If the screen appears again on my laptop. Okay. Okay, so
Roberta, may I say just a few words?
Sure.
As the coordinator of the Osteonet project, I wish to thank all of you for contributing with a very, very nice workshop to our activities. I've got to say that the two projects are much closer than we all think, because the idea of bridging the gap in knowledge and of sharing the approach among people with expertise in different disciplines is also the fundamental starting point also for Osteonet. Probably what we in Osteonet do in addition to this is trying to bridge the gap in understanding. As you were saying, when you have biologists talk to engineers or to computer scientists, very often the greatest difficulty is to understand one another and to have a common goal, not only, but to exploit the synergies in knowledge of people with different expertise. So I really thank all of you, and I hope that, since the the development of a biofabrication protocol, as well as the development of scaffolds that can be implanted to replace missing bone fragments, will require a lot more work than we think today. I hope that on the way to this goal, there will be other opportunities to interact between the two communities, between the two consortia. And with this in mind, I wish to thank all of you, and I hope that the ties will be strengthened over time. Thank you.
Thank you so much, Gerardo. I think I can speak from all of us in saying that this was a great opportunity to share our diverse perspectives and make this diversity and its value emerge from our presentations. And so thank you for inviting us. And I look forward to further interactions between the two consortia. So are there any other comments or questions from the audience? Okay, so let me thank again all of the speakers who contributed to this seminar today. Thank you. Thank you very much. And
thank you for inviting us.
Thank you.
Thank you. And I think, thank you. I think that we can conclude the seminar here, and we will follow up with the registration. And I really hope to meet all of you again and keep discussing these topics. See you next time.
Bye.
Thank you, Roberta. Thank you all.
Bye. Thank you again.
Bye.
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 See you soon.
Thank you. Bye.